Unit 2.9. This one's going to be a little bit short. We've already talked about many of the concepts here, so it'll be a nice review, kind of a good condensed place to find that information. But I'll go over it briefly. There are some videos here also. We won't watch them here, but the links are in the PowerPoint. You can watch them. I'll say a few words about them. One we've already watched in class. So one of the primary methods that we use when we are doing evo-devo is a functional assay. A functional assay is anything that allows us to determine the function of a gene. And so it's a rather simple, straightforward name, but the primary way to do that is to either turn off expression of a target gene, or overexpress a target gene, or ectopically express a target gene. Those all are ways that we can begin to tease apart the function of a gene. So if you turn off a gene, then you look to see what impact that has. If you turn off a gene, this is the classic example and is in the video, that is designed for making a flower colorful. You turn it off, you see the color gone, boom, you know exactly what that is. Now, this isn't always a simple thing to do for all genes, because some genes have a more severe phenotype. If the phenotype is death, right, so you turn off a gene and the organism dies, well, you know it's an important gene, but you don't know its specific function. So there are still some limitations to this. But even with that said, it is a very powerful way to do it. One of the primary ways to turn off expression of a gene is through RNA interference. Now, technically, this doesn't turn off the gene expression completely, because the gene is still transcribed. Now, when we say expression, in your head you should be thinking, okay, a gene needs to be transcribed. It needs to be then spliced together to make the mature mRNA. That mRNA then needs to be translated into a protein. The protein needs to be folded. Usually that happens fairly quickly and naturally. And then that protein does its job. So that entire process is gene expression. And if you interrupt that anywhere along the line, then you're going to see the impact of not having that gene around. Okay? So RNAi, technically, we still have transcription, but those transcripts are degraded by the RNAi mechanism, and you can review it in the video. There's not a lot of detail there, but that's as detailed as you need to know. Okay? The phenomenon was first observed in a petunia in an attempt actually to create a more intense color. They got rid of the color, right? So they were looking at this one gene that was responsible for a deposition of this purple color. And when they tried to insert an extra copy, they inadvertently put both the copy and its mirror image in there, so that they created a double-stranded RNA, and that's the key. Double-stranded RNA is not a natural thing in most organisms, but it is in some viruses. And so it's thought that this RNAi mechanism evolved early in the origin of eukaryotes, because most eukaryotes have very active RNAi mechanisms. You can use it in a very, very wide range of organisms. And if you inject double-stranded RNA into cells, this mechanism will be activated. Again, all the details, the molecular details of it, you don't need to really worry about. But once it's activated, then we knock down expression of the gene by continuously degrading mRNAs as they are created. So this diagram shows a little bit of some of the molecular details that are occurring with RNAi. But again, you don't need to worry about those. There's a very superficial, kind of funny level that they tell you in the video. You can get to the video by clicking on this link in the PowerPoint. So we've seen it before. I showed it in class. They do a very good job of explaining it in a simplified manner. It's enough detail to get the idea. So you can go there and follow along with that if you need to review. If you still remember it and feel comfortable with it, that's fine. But just know that RNAi uses double-stranded RNA that matches the target gene. It activates this endogenous system, or system that's already present in the organisms, to then knock down expression at the RNA level of a target gene. So we can see the impact if we turn off, or at least greatly reduce, the expression of a gene. Okay? So more details here. Again, I don't care that you know all of the details. There are actually a couple of enzymes that are involved. These seem to be very, very ancient and are found in just about all eukaryotes. Okay? So, the second and more recent method for gene functional expression is CRISPR-Cas9. This video is also kind of a little bit of a simplification. You're welcome to click on it and watch it. In fact, I recommend it. We haven't watched it in class. It goes over both the mechanisms of Cas9, a little bit of history of how it was discovered. It's kind of a cutesy little cartoon one, but they do a pretty decent job. It also goes a little bit overboard on some areas about saying some of the potential and maybe kind of hyping it, but really, I don't know, I guess I would agree that it does have nearly unlimited potential. This is going to be a major innovating force in biotechnology and in all walks of life, really in one way or another. Again, don't worry too much about the molecular mechanisms, although they go over them briefly in the paper or in the video. If you understand that level, that's more than enough. The basic idea is that CRISPR-Cas9 is a cut and paste for DNA. So we're not impacting the RNA, we're actually impacting the genetic code at the DNA level. And so we can insert sequences if we need to, so we could add genes, or we could take out genes and see what happens. We could even change small sections of genes, and that would give us a dramatic. So it's a very, very powerful tool. So not only is it more flexible in that it's not simply just, turning off expression or turning it down. We can also add a gene. You could increase expression if you knew the mechanism. So really we can do just about anything that we understand. If we understand how the DNA accomplishes something, we can make the DNA look like that to do it. But understanding exactly what it should look like to get a certain thing done might be a little bit of a trick. So it's very, very flexible that way. The other key difference between CRISPR-Cas9 and RNAi is that this can be passed on from one generation to the next. So if you do an RNAi on a really important developmental gene, it has a massive impact on the phenotype of that organism. In fact, if you choose the right gene, it might even be fatal, but it's that organism only. If that organism is able to survive and reproduce, its offspring will be totally healthy and normal because we didn't change the basic genetic code, which is what is passed on from one generation to the next. But CRISPR-Cas9, if done in the germ cells, right, eggs and sperm, or an early enough embryo that DNA then gets incorporated into eggs and sperm, it can have a permanent effect that can be inherited from parents to offspring and so on down the line. Okay, so it's a really, really powerful and a little bit of a scary tool also. And in that video at the end, they go over some of the caveats, some of the ethical considerations that need to be taken into account for CRISPR-Cas9. So watch the video and just be aware of those. Okay. The next method we're going to talk about is immunofluorescence. Now, immunofluorescence is a general term that refers to any technique where we are using antibodies and a fluorescent molecule. So that's the immuno part, is the antibodies, and fluorescence is lighting things up. And we actually review this in Lab 8. So go over Lab 8, review Lab 8. This will reinforce and go over what you're already learning there. Okay, but I'm going to do it in just a very brief overview here. So here it is in a nutshell. The first thing that we do is we take a group of target cells. Could be a whole mount embryo, could be a section of a tissue that we want to look at. And then we incubate those cells with a blocking media to minimize kind of non-specific binding. Once you block that, then you incubate with an antibody called a primary antibody. These can be polyclonal. That should be an extra L in there, so it should be polyclonal. Excuse me. Or monoclonal. Polyclonals represent multiple binding sites on the same protein. Monoclonals just represent one, but both will work fine. So we have an antibody stuck to the protein, our target of interest, and with an antibody stain in evo-devo, we are targeting proteins. Okay, once you have that primary antibody bound to the protein, you then add a secondary antibody. The secondary antibody binds to and recognizes the primary antibody, and it has a fluorescent molecule attached to it. Now, there are two reasons why we don't attach the fluorescent molecule to the primary antibody and just use it as our label. The first reason is we get some signal amplification because there are actually multiple secondary antibodies that combine to each primary antibody. So you get kind of an extra level of signal amplification there. The other reason is this gives us more flexibility. If I have some stains that are at one wavelength, I can use a separate wavelength on this, and I can just choose a second antibody. I only have to make one primary antibody, but then I can use all of these different secondary antibodies depending on the fluorescent label that I want to add to it. So we get some flexibility in the types of fluorescence that we can use by using the secondary antibody also. There are also multiple reagents that you can purchase that allow you to do direct cell staining, so we don't have to add anything or do any preps. You just add the substance and see what happens. So mitotracker red or mitotracker orange are ones that look at mitochondrial activity. So you put them in live cells, and as the mitochondria do their thing, this fluorescent label is laid down in the mitochondria, so you can see mitochondria. So here we have tons of mitochondria labeled. Another one is DAPI. DAPI is very easy and flexible to use, and DAPI binds to DNA and it fluoresces under UV light. So we get nuclei labeled very, very strongly. We can see the position of nuclei. Okay? So when we use direct cell staining in combination with, say, an antibody stain, we can get multiple components labeled. So in this one, we've got 3 direct cell staining things going on. We've got DAPI labeling the nuclei there in this blue color. We've got a reagent that recognizes actin and binds to actin, so we see the cytoskeleton, and that's labeled in red. And then we have mitochondria here labeled in green. So direct cell staining can give us a lot, but it doesn't target any one specific gene looking at expression. So if this cell was doing something differently, expressing a different cell or a different gene than this cell, we wouldn't recognize the difference using just direct cell staining. However, we could, in a combination with the direct cell staining, also add an antibody stain and see what genes are being used differentially across this tissue sample. Okay? Now, the way this is done is that we use an array of filters that filter out the very specific wavelengths that are made by different fluorescent molecules, and only let in a very narrow wavelength that matches one molecule. And so when you see an image like this, it's actually 3 different images that are all taken together. We take an image that blocks out everything except the fluorescent wavelength that the actin has been labeled with, and so you get a nice red image, but that's the only thing you'd see. We would then take a second image that filters out everything except the blue wavelength, the DAPI at the nucleus, and then we do one for the green wavelength for the mitochondria. And typically those are taken in black and white images, but then each individual image is color-coded, and then they're all overlaid one on top of each other. So it's like we're looking through all of them overlaid together. So here you can see that with just 2 colors, but we take one on the red, showing the mitochondria. We take one on the blue channel, showing the nuclei. And then we overlap them together so we can see both together. So these overlapping images give us flexibility in the ability to take images of many, many different things. And typically 3 or 4 is not uncommon. Sometimes there'll even be 5 or 6 different fluorescent labels. And so this is a very powerful and flexible tool that allows us to see many different components of developing cells. Okay? Now, to do that, you only need a fluorescent microscope. And a fluorescent microscope is simply a regular microscope with a series of filters, basically, that then you can overlap after those images have been taken. You overlap them digitally and make your final image. A different type of a microscope that we'll talk a little bit about in Lab 9 is confocal microscopy, a confocal microscope. And a confocal microscope uses a little trick of optics to help filter out of focus light. And this is critical. A regular fluorescent microscope, it can be like looking through a dirty window unless you make the sample that you're looking at very, very thin, which you can do by sectioning, but it's difficult and you can't see things in their proper position because you're only taking thin slices. So a confocal microscope is a really nice opportunity to give a better image of many different depths and different layers. And it does this by filtering out this kind of out of focus light. So when you look at an image like this, you're seeing a very small percentage of the image that's in focus, but you can't really make it out because all of the different out of focus layers that are there. Our eyes do this, but we don't notice it as much. So if you hold your hand right in front of your face and then look at something far away, you'll notice that your fingers on the hand are out of focus because your eyes can only focus on one thing at a time. Now imagine you had several different things at various lengths that were semi-translucent. You could see through them a little bit, but they're fuzzy and out of focus, so we can't see the things that we're looking at because of all these out of focus things that we're looking through. Well, a confocal resolves that, and it does it with two little tricks. The first is a little pinpoint of light. So the detector here will only get the light that is in focus because this pinpoint filters out the out of focus light, and it has to do with the way that lenses project an image onto a sensor, and they do this by basically, here's your object, we put it through a lens, and then the light rays have to cross over one another before they get to the detector. This is actually why in a camera you get the reverse image. Now in modern cameras we then automatically flip that over, but if you look at the original film where we would make the slides or the negatives for developing into films, there actually were reverse images. And so it reverses it here, and when it does that, all the light that's in focus has to pass through this one single point. So with a small pinhole, we can intercept the light that is out of focus and only get the light that's in focus. So confocal allows us to only see all the light that's in focus at one particular focal length. And then what you can do is you can adjust to the next focal length and the next focal length, and you can actually take multiple images at many different wavelengths of light, so you still have the capability of all these different fluorescent markers, and then you stack all of those in-focus images together to reconstruct a three-dimensional version of what you are looking at. So the confocal microscope is very, very useful and important. There are others that use similar principles or slightly modified ones, and lasers instead of different filtering methods, but the confocal one is still widely used and very important in Evo-Devo. Okay, so here is the end result of what you can get with a confocal microscope. So this is one where we've just stacked the multiple images, and you can get a little bit of a idea about the three-dimensionality of the structure. But then you can also, in essence, kind of almost do like in the movie The Matrix, you know, where they pioneered that camera technique. It's used in many movies now, where we move around an object so you can kind of get this three-dimensional view of what it looks like. Same thing here, and so we're putting together a three-dimensional view of these microtubules in plants, and you can see what they would look like in reference to one another. So really powerful and useful tool. Still have this idea of combining fluorescence and confocal microscopy.